Yeah, let me try to print off the pen. Okay. Yeah, you might ask, why do we need the DNA replication? When DNA replication occur, certainly for that, right, we have to talk about or have a review of a cell cycle. We know that for new cells to form, that is from if you have a cell, you want to break, divide, and form new cells, you need to have a DNA replicated. So that's DNA, we know it's a genetic material that we replicated. And it occurs in the S phase or the synthesis phase in cell cycle. So for new cells to divide, you need a DNA to be replicated first. That's in the S phase, the synthesis phase. And cell goes through the cycle, and they will divide and form new cells through the process we call it mitosis. Okay, so next slide. We know the building blocks of DNA is nucleotide. That's the molecular structure for a nucleotide. It has three parts: the phosphate part, the sugar part, and the base part. The sugar part is called the deoxyribose, or you can call it a pentose. Certainly pentose because it has 5 carbons, so that's why it's called a pentose. And for the base in DNA, it can be adenine, thymine, guanine, or cytosine. The bases can be either pyrimidine or purine. As we can see from here, purines have only 1 ring, and the pyrimidines have only 1 ring, and the purines have 2 rings. And in DNA, adenine and thymine form complement pairs, and they form 2 hydrogen bonds. And guanine and cytosine, they form complementary pairs. They form 3 hydrogen bonds. And for the sugar in the DNA molecule or in RNA, it's the in DNA, it's the deoxyribose. And as we can see, it has 5 carbons. So carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And it's deoxy, certain D we know it's lacking or removed. So deoxy meaning because it's missing this hydroxy group, which appears in which present in RNA. So RNA has ribose, and DNA has deoxyribose. So the carbon 2 position doesn't have that hydroxy group. And I guess when you think about the DNA, the very kind of common picture you come in mind is this double helix. That's DNA double helix. And if we look a little bit in more detail, we see, okay, for DNA molecules, that it is double-stranded. It has 2 strands in an anti-parallel arrangement. So we can see very clearly, right? It's a parallel because you have 2 strands parallel to each other. And the backbone is phosphate, sugar, phosphate, and sugar. The other strand, same thing, phosphate, sugar, phosphate, and sugar. That's the backbone. And the 2 strands are bound together by the hydrogen bonds formed between the bases. Adenine and thymine have 2 hydrogen bonds, and guanine and cytosine have 3 hydrogen bonds. We call these 2 strands anti-parallel. The reason is, for one strand, it runs from 5' to 3' in this direction. Then the other strand runs from 5' to 3' in the other direction. So that's why it's called anti-parallel. And if we look a little bit more in detail where hydrogen bonds are formed, so between adenine and the thymine, the hydrogen bonds form in between oxygen and hydrogen, nitrogen, and hydrogen. The 3 hydrogen bonds formed between cytosine and guanine are formed between oxygen, hydrogen, nitrogen, hydrogen, oxygen, and hydrogen. And for the one strand, here is at the 5' end, then run into the 3' direction in this way. That's the 3' position. And for the other strand, it's the 5' position here, then the 3' position here. So it's running in this direction. So it's anti-parallel. For DNA to replicate, there are many players in the process, as we can see here. Let's first get familiar with the players in the process. There are helicase, topoisomerase, DNA polymerase, primase, ligase, the single-stranded binding proteins, SSBs, the proliferating cell nuclear antigens, or the pcNNAs. And there are also concepts to understand the concept of semi-conservative replication, semi-continuous replication, leading strand, lagging strand, Okazaki fragments. For DNA replication to proceed, first you need to break the hydrogen bond which hold the 2 strands together. This is where helicase comes to play. So helicase is the enzyme to break hydrogen bond between the 2 strands. Then topoisomerase and is used to unwind the helix. And then the SSB, the single-strand binding proteins, will stabilize the single-strand DNA to prevent them from coming to wind against each other. They kind of stabilize the single-stranded DNA. Then for DNA to synthesize to proceed, you first need primase to form short RNA primers. Then they bind onto the DNA strand. Then DNA replication will occur in the 5' to 3' direction. So the same thing here, you have an RNA primer, then DNA synthesized in the 5' to 3' orientation. On the other strand, it's the same thing. Initially you have the RNA primer bound, then DNA synthesized along the parental DNA strand. As you can see, for this strand, because the DNA double strand break up in this direction, so forming a fork in here. So the DNA synthesis can proceed once it's breaking up. So it can continuously synthesize with the sliding clamp of pcNNA, and the DNA polymers will form, adding new nucleotides to the chain. And on the other strand, however, the opening up of the strand happened in this direction, but the DNA synthesis goes also from 5' to 3', so it has to proceed this way. So it cannot synthesize continuously. So you have RNA primers, then you have short fragments of DNA synthesized. These fragments are called Okazaki fragments. So on this strand, you have many Okazaki fragments formed, and then you have a ligase at the end to join the fragments together. And as a result, we call this strand the leading strand because the DNA synthesis will proceed continuously. But on the other strand, DNA synthesis occurs in short fragments. So this is called the lagging strand. This is the leading strand. The reason they call the DNA replication semi-conservative, we can see the newly synthesized DNA has one strand from the parental DNA. The other strand is synthesized new. So each new double-stranded DNA has one old copy and one new copy. That's why it's called a semi-conservative. And also because one strand synthesis is continuous, the other forming short fragments, then ligated together by ligase. So it's also called a semi-continuous replication. Okay, this is how DNA replication occurs. And I hope that you understand by now and also understand the concept in the process. Thank you.